Thank you, Mahelani. And thank you also for co-hosting with us. We really appreciate having this opportunity to come here and present. Each week will be a different talk of things that are somewhat related to the Maunalua Bay region. And so tonight I'm very excited to be able to introduce Bob Richmond. He is the research professor and director of the Kewalo Marine Lab. He has done work here in Hawaii for years. We were just talking about work he's been doing in Palau for decades. He's a coral researcher that's an expert for corals all around the world. He's been a huge resource for Mālama Maunalua in the years as we've been trying to work through restoring reefs in the area, and he's been an incredible help for us. So I'm really excited to see his talk, and I'd like to introduce Bob.
Thank you. Well, thank you all for coming out on Thursday night. And I always feel like when I'm giving a talk, it's always the students go in the back. If anyone over there wants to come over here a little bit closer, feel free to wander. I'm grateful for the opportunity to come here. My area of expertise is coral reefs and the people who depend on coral reefs. And I'll give you the punchline right now so that there's no suspense. The coral reefs are threatened, but not doomed. And it's totally up to us in terms of how that outcome comes. So what I'd like to do is talk a little bit about three things that matter to me. The first part will be context. And that is, I'm a scientist by training. It's what I'm a card-carrying member as a researcher in the Pacific Biosciences Research Center. And so a lot of my work is tied around research, but a very big part of what I do is so what? Meaning when we do science and we do the things we're supposed to do, publish in peer-reviewed journals, that's clearly something that we need to do to validate the quality of our work. But to me, especially in this day and age with the state of the reefs and the state of the world, that's no longer enough. So I'll talk a little bit about the context of so what, some of the science, including work we've done in Maunalua Bay, and then finally kind of tying it together with what the future looks like, and most importantly, what it could look like if we choose to do the right things. So without further ado, I started working on corals back in 1974. Sea level was a little bit different. Climate was a little bit different. I had more up here, less down here. But over the years, there you go, nice head. The bottom line is when I started on the left in the Caribbean, I worked in St. Croix initially and then the Grenadine Islands. Those days was 80% coral cover, luxuriant elkhorn and staghorn corals everywhere, thundering herds of parrotfish everywhere. I went back to that site about 5 years ago, and that's the upper right-hand picture. It was less than 2% coral cover. I saw one snapper, and everybody got excited because I actually saw a fish. And honestly, I went back to the hotel room and cried. I couldn't believe within my own experience over a career that's now spanned close to 45 years that anything like this could have happened to one of the most magnificent and important ecosystems on the face of the earth. I moved to the Pacific, started working in Hawaii in 1979. I was a graduate student working out of Coconut Island. I was a student at Stony Brook, but I was damned if I was going to hang around New York if I could get out of it. And so I was given an offer to work in Long Island Sound. I said nope. And so I was back on coral reefs in the Pacific. And from HIMB, I ended up going out to an interesting place I'll talk about shortly. But again, starting in 79 in the Pacific, a number of my reefs are work in Micronesia. In the lower left-hand corner is one of my reefs on Guam. And a number of them have turned into the lower right-hand side as well. So yes, there is reason to be concerned, and I certainly am, but I'm also not willing to give up the fight just yet. So here's a picture of one of the things that people do to coral reefs. And so we can talk about human disturbance. The multi-syllable word is anthropogenic disturbance. And there's a range of things we call anthropogenic disturbance. I did 2 years at Enowetok Atoll. Some of you may be familiar with that in the Marshall Islands. Was the home of the nuclear testing during the 1950s. So vaporization via nuclear detonation is kind of on the extreme side of human disturbance on a reef. And so entire reefs were vaporized, entire fronts of coral reefs fell down. The upper left-hand corner are 2 atomic bomb craters, about 1 mile in diameter, 150 feet deep. The little white dome you see there is called the Runit Dome. We used to affectionately call it the Mount Runit Recreational Area. If you've been following the news at all, there have been a couple articles in the New York Times about the dome falling apart. And this is not any surprise to any of us when you consider that the shortest half-life of one of the shorter-lived radionuclides in the dome is 5,000 years, and the half-life for concrete is 50 years. The calculation on this response was not particularly astute. So there's an interesting book I think it's out of print, but you can still find it on eBay or on Amazon, called Brighter Than a Thousand Suns. It chronicles the history of the nuclear testing program. And, we're all kind of affected by what we've seen in our lifetime. Spending 2 years at ground zero of the nuclear testing program definitely had an effect on me in more ways than one. But in the end, science is a great thing for being able to solve problems, but it can also create problems. And there's a section in the book where they talk about the previous blast that I showed you, the first hydrogen bomb blast, where they started a chain reaction, and one of the head physicists said, we're not convinced it's ever going to stop, meaning we may have blown up the world, but we're not sure yet. And so on the other side of that, a lot of the work I do in the Pacific Islands, including in the Marshall Islands, is to try to maybe make amends for some of the things that were done. But interestingly enough, if you go back to these ones, like those bomb craters, we dove in them and the corals were back and they were luxuriant. The sharks are still pretty ticked off. That's all the places I've dove. The sharks are not happy in there for some reason. But interestingly enough, this was an extremely acute disturbance. And 40 years later, luxuriant corals all throughout the bomb craters. Here are the things that look a little bit less intense on that gradient from ridiculous to sublime. Upper left-hand corner, you can see sedimentation from runoff. If you're in Hawaii, this is typical. Maunalua Bay, Kuli'o'o, Wailupe, any of the sites, this is what you see all the time. Right-hand, upper right, overfishing, especially of reef herbivores, the lawnmowers. Lower left-hand side is a sewer outfall on Guam. And I'm finishing up my coffee table book on the sewer outfalls of the Pacific Islands. It's bound to be a bestseller, so keep your eye open for it. But I've dove every single outfall in Micronesia except Coast Rai. When I was there, the waves were too big. So they keep asking me when the weather's good if I could come down and dive their sewer outfall. It's a freshwater effluent. It surfaces 100% of the time. Due to federal EPA legislation, it has to be in at least 60 feet of water, which off of New York is 2 miles offshore, which off of Guam is less than 40 feet offshore, meaning you can throw a rock from the edge of the reef into that plume. What you can see are particulates. What you can't see are the variety of dissolved materials that are in that, including things like unmetabolized birth control pills that pass through women with a very large estrogen signal. And fish and corals used estrogen to be able to cycle for reproduction. So we're loading up the water with endocrine disruptors, endocrine chemicals. I'm sure you've all heard about the sunscreens. So that's been a hot topic. If you look in your bathroom, I run an ecotoxicology lab down at Kewalo Marine Lab. And I say most of you in your bathrooms have probably more toxic chemicals than I keep in my ecotoxicology lab. Look at your shampoo bottles, look at a variety of things that are there. Personal care products are a big issue, and it's starting to finally get people to understand it. But sewer outfalls are an amazing area for a variety of chemicals getting into the environment and wreaking havoc on populations of organisms because of what happens there. And then finally, recreational impacts. I'm sure you've seen it on Maunalua Bay as well. Compression waves from jet skis. Whoever came up with these jet packs, they pump 1,000 gallons of water a minute. The ultimate wearing blender for any plankton, larva, fish, you name it, it gets in there, it's ground to shreds. And so we've been working with Fisherman's Hui in Maunalua Bay, advising them on the fact that one of the channels in which these people are operating was a spawning channel years and years ago, and this is no place to be operating these kinds of vessels. So this is one of our 2 rescue dogs. His name is Momo. He's a Aussie Shepherd, born to herd. That's what Aussie Shepherds do. We rescued him when he was 6 months old. And so I live in Kaneohe and Kahikili in the morning on Sundays. There's this constant flow of buses going up to the North Shore. And so when he saw the first group of buses go by, the herder that he is, he looked at me and he goes, I got this. And it's like, no, you're not going after the bus. And so, it's if it moves, it has to be herded, but I'm not going to let him herd buses. And just again, for the context, there's a whole bunch of us that kind of feel that same level that we definitely need to start herding decision makers. And I wonder if we have about the same chance as my Aussie Shepherd does with a bus. But there's a group, including groups like the Aldo Leopold Fellows, who are specifically trained on how to work across politics, across decision makers, state, federal, international level. And I sometimes feel like my poor dog wanting to take on an impossible task, but persistence beats brilliance. That's how I got where I am, and I'm going to continue to try. So we go to DC about 4 to 5 times a year and keep plugging away. And my final layout for what I'm doing here is the science to policy bridge to me is just so important, and it's hard to explain to people. So for Hawaii, this is easy. What's that? Everybody knows. spamasubi. So to me, spamasubi is the ultimate analogy for trying to bridge science to policy. To me, the science is the rice. It's granular. It's made up of lots of solid things. It's a solid base upon which there is no spamasubi without rice. To me, the egg is kind of the management part, the practical part. It's solid. It's protein. To me, the policy part is spam. Nobody knows what's in it. If you ever look at what it is, it's meat byproduct. I don't know what a meat byproduct is. But nonetheless, you can't have spamasubi without that magical layer who nobody quite understands. And to me, that's what policy is. I don't understand it, but you can't do what we need to do without good policy, including good legislation. And the good news is we just got legislation to review on protecting coral reefs for the next 20 years. And it's going to be introduced in Congress next 2 weeks from now. And so there is hope. We're just trying to make sure it's good stuff. So with that prelude, which has probably confused all of you, what I really work on is the ability of coral reefs to persist over time. And it's made up of 5 integrated steps, starting with reproduction, the formation of new individuals from prior stock. If new coral larvae, coral babies, coral seed, whatever you want to call them, are formed, they have to recruit. That is, they have to find a good place on the reef to settle, metamorphose. They need to attach. They need to become the generation of corals. If they survive, and as many of you probably know, corals are this really cool thing. When you say, are they animal, vegetable, or mineral? The answer is yes. They're animals, but they have vegetables. They have zooxanthellae, little algae that live inside, and they're also mineral because they make calcium carbonate. So many of them have to pick up their symbiotic algae. They're basically solar powered. Most corals get 90% of what they need on a daily basis from the sun if they pick up the right kinds of algae. And so that's a very important part of them. If all goes well, then they can grow, and eventually if they mature, they can go back to reproduction. So the 4 pictures there, upper left hand is a daily spawning event. The right hand, upper right hand, is a coral recruit, uptake of zooxanthellae, and then finally growth. And so these are completely dependent on water quality, bottom quality, stressors that can be a variety of both biological and abiotic, and then the interactions that occur in terms of the interventions we may be able to do. And more specifically, this is a daytime spawning of a very common coral here in Hawaii, 7 o'clock in the morning, 3 days after the summer full moon, for about 5 minutes. And what we found was when this coral releases its egg and sperm into the water, and that cloud drifts down to the next coral of the same species, that one will go, then the next one will go, the next one will go. And it may not matter exactly which night they pick, although it is tied to nocturnal illumination and temperature cycles, but it is critically important that the same species go off at the same time. Otherwise, there's no hope for getting new corals and new coral babies to come back. We call this a contagious spawning event, and it shows that these things are chemically communicating with one another. When they pick up the right estrogenic signal from one colony, it's enough to cause the next one to go off. If all goes well, in some similar corals, we see these clusters of egg and sperm that are released together. This is a nighttime spawning of corals like Montipora, very common here in Hawaii. Acropora, less so here, but more so in the Western Pacific. And oftentimes what they do is bundle their egg and sperm together. They would do it for a reason because the eggs are full of lipid, they float. And since corals are at various depths, the corals can't interact with one another. But if they release everything and it comes up to the surface, then the egg and sperm can interact, which is what really matters. And the problem is because they use this, it works really well over evolutionary time, the egg and sperm float to the surface. If there's a rainstorm at night, fresh water is lighter than seawater. And when you have runoff from land, it can contain pesticides, it can contain hydrocarbons, it can contain heavy metals. You get the most sensitive cells, gametes, egg and sperm, hitting this upper layer. And if it was a rainstorm on the night of spawning, you may lose everything for that entire year. And most corals only go off for about 20 minutes, once, possibly twice a year. And so if you have a bad event due to a rainstorm or some runoff, then you're done for that year. And we've been able to document on a number of Pacific Island reefs reproductive failure year after year after year, in some cases up to 5 years in a row. If it does work out, eggs are a single cell, and when they're fertilized by the proper sperm, they begin to develop into an embryo. If all goes well, that embryo will end up becoming what we call a coral planula larva. It's just a coral seed. And then at that point, they need to begin to search for a place to settle. One of my students did some great work back in the 1980s questioning, do coral seeds simply get randomly dispersed with currents? And if they picked a good place, they live, and they pick a bad place, they die. And it turns out that they're actively selecting for the bottom. Their entire bodies are basically rolled up with taste buds. They come down to the bottom and they lick. And what they're looking for are either a certain kind of bacteria or a chemical cue that's associated with a very specific kind of coral and algae. If they don't get that, in this picture, we had a piece of rubble that had only a tiny, tiny spot of the right inducer, and that larvae swam all around and found that one spot. Had that spot not been there, that larvae never would have settled and would have eventually died. And then, as I mentioned, for many of these corals, they don't have their algae passed on through the egg or the sperm, but they have to pick them up after they settle and metamorphose. So the point I want to make here is everything you're seeing up there is chemically mediated. The first 4 stages completely by water-soluble compounds, and the last 2 by what we call lipophilic fat-loving compounds that dissolve with fat rather than water. And so the problem is that you can have successful spawning, good fertilization, embryological development, and larvae forming, but if they can't get the right metamorphic inducer or they can't pick up their zooxanthellae, it's as if nothing happened whatsoever. And you can only do that so long until the reef, corals are living creatures, so they can die any year. And if you have multiple years of reproductive failure or recruitment failure, then you know your reef is going into decline and it won't come back. And this is an example. No coral larva could possibly settle here because they don't get their inducer. They can't settle on sediment. They can't settle on soft algae. And so this is one of our sites in Palau where I just came back from. The good news is this was taken 10 years ago. That site now, because of work that they did to improve their watershed management within the watershed, storms have lifted up the sediment. They've got their herbivorous fish back that have eaten the algae down. And now we have baby corals everywhere. So it was good to be in Palau and see that when you do actions, you can switch that downward trajectory into an upward trajectory, and it doesn't take much. Within 10 years, we've seen substantial recovery of that reef. So the typical tool we use for assessment and monitoring coral reefs are things like quadrats and transects. Sometimes we use videos, but it's basically PVC frame with a bunch of strings on it, and you count. The problem with the traditional approach has been you're using depth as the indicator of stress. And to put it into human terms, I know we all have stress in our lives. People don't realize being at university can be stressful. And so if I come home after a stressful day and my wife said, "How's it going, dear?" And I said, I'm feeling kind of stressed, and I fall over dead, chances are I should have intervened a little bit earlier. So what do we do with people? We do blood pressure, we do urinalysis, we do a variety of things like that, cholesterol tests. And so you can see if you're starting to get stress markers coming up, then you can act accordingly. So if my doctor says, "Hey, Bob, your cholesterol is 280, that's not good. Let's go ahead and do an intervention. Let's go ahead and cut out the beer and pizza, run 3 days a week, come back in 2 months." And if I go from 280 down to 210, she'll say, "Hey, that's good. You did an intervention and you got lower, but we're not there yet. Let's get another 30 points. So at least I have something to go with." What we typically do with reefs is you're alive, you're dead, and we know that there's a lot of stuff in between. So this is basically the philosophy of our laboratory. Mortality is a rather crude estimator of stress. And you can see that reflected in modern politics, that if we use the category of alive versus dead, there would be no argument about Obamacare, period. You don't need health insurance, you're either alive or you're dead. We know that's not true. So here's my childhood hero. I see a few people here who might know who the guy is on the left. Who's the guy on the left? Rex Harrison, and he was the original Dr. Doolittle, okay? And the younger generation knows the guy on the right is Dr. Doolittle. I understand they're first cousins once removed, but the new generation doesn't even know either one of them. That's so embarrassing. I just did a talk at a school, and they're like, "We don't know who either one of those guys are." Our children today are deprived. They don't know who Dr. Doolittle is. So what was it so cool about Dr. Doolittle? What could he do that nobody else could do? Come on, you all know the song, "Talk to the Animals." So again, I have to clarify. I have the 2 dogs, 2 rusty dogs and a cat, and I talk to them a lot. Don't chew on the cat. Put down my Zori. I talk to my dogs. It doesn't matter. So there's a difference between talking to something or listening to. For those of you that are married or in any kind of relationship, you know what I mean. Talking to and listening to, different. So it was this concept of, could we actually listen to animals the way that Dr. Doolittle did? And the answer is yeah. As a matter of fact, a lot of traditional cultural practitioners in Hawaii and throughout the Pacific know it really well. Some of you may be familiar with Rachel Carlson's incredible book, Silent Spring. And people know from Audubon Society, you can go out into the forest. Eddie Kamai, locally, a lot of people know the work he did, Listen to the Forest, was a brilliant piece on going into the forest and being able to know what's there simply by listening. You can listen for birds, you can listen for insects. And Rachel Carson's point was, if the spring starts and you don't hear birds and you don't hear insects, she tied it to a variety of insecticides that were doing damage to the community. But we can do the same with coral reefs. And I actually work a lot with traditional fishers who can go into the ocean with their eyes closed and they can tell me a lot about the state of the reef within 100 meters without even opening their eyes. They can tell you which fish they're hearing. A good coral reef should sound like downtown Manila at rush hour. It should be this cacophony of noise from shrimps and crabs, parrotfish biting corals, grinding with their pharyngeal mills, and being down in Palau teaching a bunch of students, I would have them do it. We're going to go out to the reef. First thing you do is get in the water and close your eyes and listen. And every site we went to, they started to pick on the fact that they could tell the difference between all the sites just by listening alone. There's some really cool papers, and if you want to Google it, you can actually get the sound of a live reef versus a not live reef on Google. So the internet is helping as well. So here are 4 what I call Dr. Doolittle moments. The upper left-hand corner is not a chia pet. It was a living coral, and it's telling me in this ecosystem, too many nutrients and not enough herbivores. The lawnmowers are gone. The herbivores have been fished out. Too much runoff and sedimentation with nutrients. So that coral is covered with algae now, and it's dead. And this reef will never, ever, 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 ever recover until we shift that back into a system where the substrate is appropriate for coral larvae. So the 2 things we look for: is there a supply of larvae, A, and B, if the larvae come in, could they actually settle? And the answer to panel number 1 is no. Number 2, you can see that kind of little red bar in the center of that coral. It's a very common coral here in Hawaii, the lace coral, Poeciloperidama cornus. It always has symbiotic crabs and shrimp that are associated with it, and it's a great relationship again. Crown of thorns starfish eat coral. They love to eat this coral, except if they have the crabs and shrimp. They are the bouncers. These are the security guards. They actually make little bags of lipid to feed them and provide them a place to live. If a crown of thorns tries to crawl onto this coral, the crabs and shrimp will immediately come out and begin to tear away at the tube feet in the stomach of the crown of thorns starfish. And if you've never seen a starfish gallop, you're missing one of life's great, wonderful things to see. Crown of thorns can actually move really quickly when they want to. And if they come up on this coral and they start getting attacked, they move really quickly. So this is another Dr. Doolittle moment that if I see this coral, and because I do so much work with traditional communities of fishers and traditional gleaners, they know the coral, they know the crabs and shrimp. I ask them to go around and take a look. If they see the coral but not the crustaceans, I know that it's likely a pesticide issue. The reason why crabs and shrimp are very close relatives of cockroaches and ants. And if there are insecticides in the water, the crabs and shrimp will die way before the coral will. So in many of the things we do, I don't have the funding to bring in sophisticated LCMS and do a lot of water quality samples, but you don't have to. This is another Dr. Doolittle moment. If those crabs and shrimp are missing from that coral, 9 times out of 10, it's an insecticide issue. Lower left-hand corner, these feather duster worms, we call them sabellids or sapulids. They can only live in areas with a lot of particulate organic matter, which is tied to sewer outfalls and nutrients and runoff fertilizer. If they start showing up, we know that there's a problem, and that's what happened to Kaneohe Bay. When the outfall was inside of the bay, these things were everywhere. And a lot of what we call boring organisms, not because they're not interesting and fascinating, but they undermine, they bore right into the calcium carbonate, make it porous like termites to wood. And after a following storm, large boulders of corals broke off and collapsed because of all the boring organisms. And then the last one I showed you is the coral larval recruit. It picked the right place. We look, are they on an upper surface or on the side surface or an undersurface? If they can settle metamorphose, that's one story. Number 2 is, do they settle on upper surfaces? That's a good sign. If it's vertical or undersides, that means you've got a sediment issue in hand. So all of these are Dr. Doolittle moments that can help us understand what the reef is doing. And if you want to know the state of a reef, ask the corals. So Maunalua Bay, this is where we pioneered a number of these studies. You can see Hawaii Kai, there's the marina, Port Lock off and to the right, and then we have a number of flags there. We started to develop these new tools called omics, genomics, proteomics, transcriptomics, and metabolomics. And these are molecular tools that you can use to try to parse out the same way we would use a blood test or urinalysis for a person. We can now do this with corals. And so it turns out that corals have the exact same proteins people do. And if you listen to the commercials on TV, Lipitor or any medication will say a simple blood test will tell you if it's safe. They have to say that by law. And if you take a new medication, normally the doctor says, all right, come back in 3 days and take a blood test. What they're looking for is your liver enzyme. Bilirubin will go up if you're having a toxic reaction. So your liver is kind of your oil filter. And that's the place of response, that if you're taking something and having a toxic reaction, your liver starts to pump out detoxification enzymes. A little digression here, but learning science as a kid, I hated liver, and my mom used to feed it to us. At 7, I started getting interested in biology, and I understood what a liver did. And one day when she tried to feed us liver, I went off on her saying, my God, woman, what are you doing to us? Would you feed us an oil filter? Why would you do this to the children you pretend to love? never had to eat liver again. So having science as a background is good at an early age. But we use the same knowledge that if livers can do this for people, even though corals don't have livers, do the other cells do it? And what's cool about this is corals are notorious liars. They lie about their taxonomy, they lie about their age because they can grow and die back, but they can't lie about basic protein expression. And so this is a kind of analysis called canonical correlation analysis. Don't worry about that part. But look at the circles, what we call centroids. And what you're seeing are 3 sets of sites. Site N is the one that was closest to the mouth of the marina. Sites 3 and 2.5 were midway out of Maunalua Bay, where there's a lot of invasive algae. And sites T and K were right along Port Lock on the outside. And then the one for Maui is a reference site to La Perouse. If you know what's out there, there's nothing really at La Perouse. It's a pretty clean site. So what we did was query the corals. This was our Dr. Doolittle moment. We took little tiny samples and analyzed the protein expression. And what we see is that closest to the mouth of the river, the further you are away from Maui, either to the right or up, is showing stress and stress proteins being released. The one at site N is showing what's called MXR. It's a protein that only gets expressed when you're getting exposed to toxicants, oil, heavy metals, pesticides. That's what happens. Sites 3 and 2.5, there's a kind of protein called an SOD, a superoxide dismutase. That is made when you start to go into oxidative stress. That's an area where there's a lot of invasive algae, a lot of the grassolaria. We saw that in the areas with the mudweed. Remember the big mudweed issue, the great hoopie? We were getting superoxide mutase because during the day, plants photosynthesize. But at night, they suck the oxygen down, and these animals go into oxygen deficit and begin to suffocate. So what do? They make proteins to prevent them from suffocating, and that's what SOD is. It really surprised the heck out of us. I was expecting further out that we wouldn't see any effect of human activities, and we were getting GPX and MUT-Y, which are indicators of DNA damage. And so I'm trying to figure out for the life of me, why are corals along Port Lock on the outside showing DNA damage? And so I went and talked with the guys at Mackay Watch, and I said, what is it about Port Lock? And I was saying, well, those are $1 million homes. Maybe they're using a designer termiticide when they tent. And he goes, I don't know, but everyone there has a swimming pool, and when they drain their pools, it all comes down into the drains and all comes down at the ocean. And that was the indicator. Chlorine and bromine cause DNA damage. And so what we think is happening, this was the smoking gun for DNA damage caused by these chemicals. The good news is they'll volatilize. And if you put the stuff aside and you let it volatilize off and don't dump it in the ocean, then the corals won't be suffering DNA damage as a result. So what's really cool about this is in the past, everyone's pointing fingers. And Maunalua initially was a group of people saying, let's go ahead and make Maunalua Bay a large marine protected area. Stop all fishing and everything would be fine. And that's when I got dragged into it. One of my good buddies from NOAA, Jerry Davis, who does MPAs, and we worked together in Guam for 20 some odd years. He said, hey, Bob, have I got a deal for you? Whenever he says that, I know I'm in trouble. And he said, we need science. We need scientists to come in. Everyone was blaming the fishermen, and the bottom line in Maunalua Bay is you could have stopped fishing immediately and it wouldn't make any difference because you have 9 subwatersheds dumping tons of sediment and a variety of discharges of pesticides, insecticides, oil, compounds, you name it, was coming in. And so we began to talk with them saying, well, why don't we ask the corals what's going on? That's why we started this original work. And this showed exactly what was going on from near shore, midshore, and offshore. We could show what was pollution exposure, what was invasive algae, what was pool chemicals. And inshore, you can see in the upper right-hand corner, high turbidity, but there are corals that are living there and doing just fine. Further out, the water's pretty clear, but they were still suffering from the things that you couldn't see that were dissolved in the water. So having these new techniques have enabled us to do things we could never do before. And what's really cool about it is just like with people, this is a graph showing the amount of protein that was expressed over distance. So on the left-hand side is the relative amount of protein that's being upregulated. And then as you go from the left to the right, Maui is the first one, and then you go from near shore to further midshore and then to offshore. And we call this dose dependence. And this is the cholesterol test. That's your 280. You start to do diet and exercise, you get down to 210, you're doing much better, but we really need to get back towards Maui. So 2 things that the managers asked us. Can you show us the smoking gun or smoking guns when you have multiple stressors? Answer is yes. And number 2, can you do it in a way that enables us to determine if we're doing something that we're actually getting some return on the investment? A lot of our focus now is looking at these threshold levels because these proteins often have dual purposes. Interestingly enough, the same protein that does detoxification is the same protein that's tied to reproduction. So if you're using all that protein up trying to protect yourself from gasoline and insecticide, then you don't have anything left to go into reproduction. So there's a trade-off there. And now we can actually ask the corals not only what's bothering you, but at what level. And now we're beginning to get those threshold values that we can use to inform managers. So this is very self-explanatory. I won't go into any detail here. We call these our subway maps, but this is like the coolest thing in the world. You may not think so, but I do. What these are in the 2-dimension is everything that goes on in any living organism, whether you're a coral or a worm or a slug or a reptile or a bird. These are things that if you've taken biology, Krebs cycle, mitochondria, everything from how do you metabolize, how do you make proteins, how do you reproduce, everything else. And what we do is take the proteins and using this really cool thing called liquid chromatography mass spectrometry, we can throw these data up on this chart, our subway maps. And what you're looking at here is a really, really cool experiment that one of my past PhD students went on to be a postdoc, Kaho Tisthammer, did, where she took some corals from Maunalua Bay that were outside, moved them in, and took some of the corals that were in and moved them out. We call that a common garden experiment, reciprocal transplant. Everything that you see that turned bright blue there were proteins that were turned on when the corals from the outside were brought in, meaning that even a short distance of about less than half a football field was enough to freak these corals out. They were doing okay outside, you moved them in, and they just started switching on everything on their circuit breaker for detoxification, oxidative stress, DNA damage. You just go right down the list. And when she took the corals from inside and moved them out, they were in heaven. They were doing their yoga poses and doing their ohms, and they were just happy. A little bit of green here and there. They were just tweaking it. They were just kind of saying, no, we don't need this one. We'll turn that one out. We don't need that as well. And once again, the corals can show us using proteins what's going on, what's bothering them. And over these very short distances, that surprised the heck out of us. So what we can do in the laboratory, not only measuring corals in the field, but we can actually do very selective exposures. This is one of the experiments she ran using PCBs, atrazine, which is very commonly used in Hawaii as a pesticide. It's a really nasty one. This is the one that causes feminization of frogs. So male frogs now have ovaries. That's not good. And then Roundup, glyphosate, which has been in the news lately. And so what she was able to do is to take corals and expose them to very, very low concentrations in the laboratory, then look at how many proteins were upregulated, 99 for PCBs, 175 for atrazine, 5 for Roundup, and then look which ones were the same. And you can see there's a little bit of an overlap between PCBs and atrazine, 23 of them. And when we go back in, we can see the ones for detoxification, we can see the ones for reproduction, we can see metabolism, we can see DNA. So we can really do CSI or sophisticated medical intervention, finally, for knowing what the corals are doing. And using the ability to look at the amounts that are there, we can actually write a prescription for the coral, or more importantly, for the people who are affecting the coral to say, these are the things you shouldn't be doing, these are the things you should be doing, and then we can watch the corals as they get better or worse. So in addition to Maunalua Bay on the right side, those are the inside and the outside. That's where Kaho switched the corals back and forth. The other one on the left-hand side is West Maui. So we have sites at Honokauai and Waihekule are the two that are there. We also have Polonui and Olawalu, if you know those sites as well. And so we began to ask the question, if we're doing all these things and the protein expression is changing, I wonder if we're getting different genetic problems going on out there. Meaning, some of you may be familiar with what was called the Irish potato famine. The Irish potato famine was not a potato issue, it was a genetic issue. There was one kind of potato that grew really, really well under very harsh Irish conditions, very cold, very dry, very poor nutrients. So they stopped growing different kinds of potatoes and grew that one genetic variant that did really, really, really well under those conditions. The problem was that one genetic type did not have any resistance to the pathogen that came through and took everything out. 1 million people starved to death, 1 million people immigrated. It was a worldwide issue. And that was 2 million people, which is tragic. There is an expected 100 million people worldwide that depend on coral reefs. So what we are approaching now is what we call the Irish potato famine of coral reefs. When Kaho compared the genetics from inside and outside in Maunalua Bay, they were genetically different. We lost genetic types inshore. We had more genetic types offshore. So the ones that are very resistant are doing fine inshore, but we're losing a whole lot of sensitive ones. Same thing in Honokauai and Waihekule when we did the pairs. Outside always had more genetic diversity than inside. But what surprised the heck out of us is when we compared the inside populations at all three sites with their outside populations, they were much more different than they were to all of the inside populations, meaning that the population inside of Maunalua Bay is more closely related to the inshore populations on Maui than genetically they are to the ones that are 50 meters away, 50 yards away. And that to me is the scary part. We're losing genetic variation and we're not even paying attention to it because it's hard to see. We finally have the tools to do it, but that's the warning sign. That's the red flag that's telling us we're losing genetic diversity in a world where we don't know what the next stressor is going to be. We have corals that do very well under high temperature, so everyone's saying, let's go ahead and grow all of those corals. Then all of a sudden we have a disease outbreak. And when we compare the genetic variant that does well under high temperature, it does really poorly against disease. So that's one of the things we're very concerned about when we talk about these kind of selective breeding. I'm lazy, so I just go to ugly places. And so we've been doing work in Honolulu Harbor. Remember Matson molasses spill? We got called by William Isla at 7 o'clock in the morning. Somebody's got to come down and help us out. We got to find out what's going on there. There were amazing reefs inside of Honolulu Harbor. They shut down all the traffic for the day. A couple of my students went in, we got the corals, and I never would have expected any corals to be in there. And there were beautiful reefs inside of Honolulu Harbor. No self-respecting coral should live in Honolulu Harbor, I can tell you. You have all the urban runoff. You have anti-fouling paint. You have gasoline spills. It's just nasty. And there were luxuriant corals in there. But when you dump a bunch of molasses and the phytoplankton blooms and oxygen goes to zero, and the sugar water creates osmotic pressure that causes the cells to explode, the corals didn't do really well. But when we go in there, we have genetic variants, and now we're in discussions with the military to say, Pearl Harbor, you've got corals there, you've got to move. We'd really, really, really like to do our chemical Dr. Doolittle science with them. And now they're getting excited. At first they were worried that you guys are going to figure out what we're doing. So that's not our goal. Our goal is you probably have some of the most valuable corals in the world. And when you're trying to get permits, you can always tell NOAA, we have these incredibly valuable corals. We'll trade you some of those in return for something back. So we're in discussion. So we'll see how that goes. But we can now use these tools to answer some really cool questions. So let me talk about context again. This is not actually what the Pacific looks like. That thing is the United States superimposed, but that's where I do a lot of my work still. So Hawaii is in the upper right-hand corner. I lived in the Marshall Islands for 2 years where I spent time working on the coral reefs there. I do a lot of work in the Federated States of Micronesia, Kosrae, Pohnpei, Chuuk, and Yap. I was on Guam for 18 years at the University of Guam Marine Laboratory until my lovely wife said 18 years on Guam is sufficient, dear. Where are we going? So we came back to Hawaii. CNMI, Commonwealth of Northern Marianas, and Palau, where I was just down for 2 glorious weeks. And so these are also areas that have a fair amount of interactions with their reefs, cultures that are very much tied to them, but now coming against all the impacts of human development. American Samoa is also part of our group. We have an educational grant, a workshop starting tomorrow with people from all of these island groups. And so I've had the chance to work in these islands for decades. So this is in the upper left-hand corner is Maupialug's home village on Satawal. I was asked to go down and help the Council of Tamala when they had a huge ship grounding on Satawal. So I got a chance to see Mau's community building a new sailing canoe. I tried to charter it back to Guam because we were on this horrible vessel with rats and cockroaches. And I said, can I charter this? He said, well, it's about 2 more months until we're done. And so I had to wait, go back on the other one. Below the sailing canoe is a breadfruit kite. They have some of the most sophisticated fisheries management systems I've seen anywhere in the world. The breadfruit kite is made with breadfruit, pandana spines. They make sennit rope. They fly the kite over the reef, and then they pull a line. The sennit rope comes down. They don't use fish hooks. They use muscle bands from oceanic sharks. They make loops. They dance them on the water, and the only fish they can catch that way is a long-nosed needlefish. So the needlefish grabs it, the snout gets caught, they pull it back in over the reef, no bycatch whatsoever. And so each different community or each different caste has their own way of fishing, and it's the most sophisticated system I've ever seen anywhere. A few years ago, a couple of my dear colleagues from NOAA, who I love, they're great friends of mine, but this group came like, we need to teach these islands how to use Magnuson-Stevens Act to protect their fisheries. And so Magnuson-Stevens Act enabled the collapse of fisheries off of New England, off of Florida, off of Texas, in the Gulf of Mexico, off of California. And so the great thing about Micronesia is you can call a few people and say, don't let them get anywhere near you. And for 2 weeks, they played whack-a-mole. Oh, yeah, we'll be happy to meet you. And then they never showed up. And so these guys called me up and said, I don't know what it is out there. We made appointments. They didn't show up. We couldn't find anybody. Everyone was gone. And I was like, phew. So, we kind of gave them the heads up. But in the end, they've got this incredible, sophisticated, thousands of years of history of being sustainable. The problem with traditional ecological knowledge alone is it never lived in a world of climate change at the rate we're seeing today. They didn't have to deal with things like organophosphate pesticides. So we need to marry the best of traditional ecological knowledge and cultural practices with the best of modern science if there's any hope for the future. And so it's kind of a tale of four cities: Guam, Palau, Pohnpei, and Hawai'i. Upper right-hand corner are the Rock Islands of Palau, fantastic. Lower left-hand corner is one of our sites in a watershed discharge in Pohnpei. Here are two of my fishing buddies from Guam. And so in the case of Guam, they were protecting a coastal area, but there was a road project up on the road that was causing sedimentation from a fairly small watershed, about 4 square kilometers. You can see a little river discharge, and this is kind of an animation showing near shore all the corals were just buried and killed. A little bit further out, corals were kind of a few of them alive, but they were dying. The next circle is a few corals were kind of holding steady but not growing. A little bit further out, they were growing but not reproducing. Next circle, corals are actually growing and some eggs and sperm started to form, but they weren't good enough to be able to be successful. A little bit further out, the egg and sperm were being produced, but when they floated to the surface during the summer, during spawning, they hit the runoff from the stream, and so they were killed instantaneously. A little bit further out, there was some fertilization that was okay, but the larvae couldn't settle there because of the sediment. So what we saw was that the ocean, that the land was pushing the reef away from land. Each one of those concentric circles. So the issue here is coral conservation begins on land. Maunalua Bay example, perfect. And whatever you do on land today ends up in the ocean tomorrow. So by concentrating our efforts within the watershed, the goal was to move things back. It's a small community there that has about 65 families. There were a couple of hunters that were lighting fires to burn, so it was easy for them to hunt deer and pig. And everybody in the watershed knew it was Uncle Mike. So we went to the school. There are about 300 kids in the school, and we found this works really well. That anyone who's been a parent or a grandparent or been around kids, they are the ultimate vector of every disease known to humankind. They go to school, they get sick, they bring it home. So the model was, well, if they can carry disease, they can also carry environmental ethics. We can also use that as a way of getting back to the people who really should know about this stuff but would never go to a meeting. So we got the kids fired up, and we find kindergarten, first, second, third. That's like primo. Get them wired up, send them home, and let them loose on their families. And you're never going to win an argument with a 5-year-old on a mission. I'm a dad. I know all about this stuff. And so we went to the school. We started working on watershed rehabilitation. The guys from Aquatic Resources and Wildlife Resources said, well, we can stake them out, we can arrest them, give them a $200 fine, throw them in jail, let them go. The next day they're going to start another fire. But when 300 kids went up to Uncle Mike and said, he never lit another fire again. So again, working through communities, that there are a variety of techniques, and community-based peer-to-peer or youth to adult is actually a good way to go. And in the case of Guam, that worked the best in terms of stopping it and then getting the hunters more engaged in other alternatives, including then the government said, well, if you're trying to attract them, we'll just give you salt licks. You don't have to light fires and burn the land and cause all that damage to the reef. So one of my past students worked on sediment socks. This is not the world's largest anaconda, but he worked in this watershed, and this was showing May 2012, July, and December. He's Chamorro, one of our PhD students from PBRC, and he's now the director for Sea Grant at the University of Guam. So this is a success story. Worked the community, and you can see in less than a year how much restoration, and part of his PhD was calculating how much of that watershed they had to restore using how much sediment sock to bring that threshold below the area that was affecting the corals. And now the reefs are coming back and the fish are coming back as well. So the fishermen have taken a very active role in leaving the herbivores alone. We said, take all the snapper and grouper you want, carnivores are fine, but we need the lawnmowers. So they've been doing a great job. And thanks to Austin Shelton's work, it's a very good success story of what you can do through a community. Here's Palau showing when they were starting to fill and grade mangroves. Mangroves are very important in Palau. They're not in Hawaii. Well, they are here, but they shouldn't be. And so mangroves are a problem here. So I'm not saying we should have mangroves because we shouldn't. But in Palau, it's a very big part of their area, and they trap about 30% of the sediment. So they were cutting the mangroves to build new housing areas because that's quote unquote government property. It was easy to lease it. And as they were filling in the mangroves, you can see in the upper right-hand corner what that was doing to the reef and the fisheries that were associated with it. We actually went in and did our Dr. Doolittle science, and those 3 circles there, we measured corals from one area that was high sediment. That's the one in green. We did one for an area that's really interesting. It's a bay that's been set aside due to sea level rise for about 5,000 years. And the corals in there are genetically different than anything we've ever seen. They're living at about 32 degrees centigrade and a pH of 7.9, which is what the predictions are for the year 2050. Now it may be 2030. But again, it was like, no, corals shouldn't be able to live and grow there. They're just luxuriant reefs there. And when we asked them how they do it, their protein patterns are completely different. And then the one in blue is this beautiful reef area with, raging currents, crystal clear water, and tons of fish. And what it showed us was that each one of these populations had a different protein pattern, and that reflected what was going on there. And then when we did the genetics on those, it turned out that even though they're the same species, the one that we get here, that big massive coral, turns out they're completely genetically different to the point where we now would consider them different species. So what's good about that is that corals can adapt genetically over time. We know that they can play the games with the proteins, and 3 areas that are totally different, the protein and genetic patterns are completely different as well. But it's giving us hope to say, we know that there are genetic variants out there that can do phenomenally well under different conditions. And if we can tap into that, either through selective breeding or there are other tools that we can do, there's a chance of using the science to help ensure that there are reefs for future generations. So here is again this wonderful, beautiful subway map. And this was showing what happened to the corals in the areas with this scenario for 2050. All the red are the proteins that they're turning on, but it's working. They're actually surviving, they're growing, they're reproducing. And the way I look at it, it's kind of like a bilge pump in a boat, that if you have water coming into your boat and your bilge pump turns on, as long as the water coming in is slower than the water going out, the boat stays afloat. And that's exactly what they're doing with these proteins. That's their bilge pump. So even though they're being expressed, that's not a bad thing. It shows that the system is working. And what we're able to do is track which of the proteins that they're doing it. And at this point, they're still keeping the boat afloat. There is a point where that threshold will be passed, and that's what we're trying to understand. But for now, just the expression of the protein is not bad. It's actually showing us what they need to be doing in order to stay alive, and that can help us figure out which of the corals we may want to selectively breed or transplant into the future. The other thing we do is work with communities, local cultures, and so for a solution to work, it has to be effective, it has to be economically feasible, and it has to be culturally acceptable. So because the work that was going on in the watersheds was causing all the sediment and they were cutting down the mangroves, taro is a very important crop for the people of Palau, like it is in Hawaii, and that food security is a very big issue for them. They're like Hawaii. Every 2 weeks they have ships coming in with food, and if the ships stop, they're out of luck. There's not enough taro grown in Palau to be able to support their populations anymore. So we began to work with some of the women's groups that do taro, and we started to modify the taro fields a little bit with those 2 trenches to catch water and catch sediment. And what we found is where mangroves were 30% effective in trapping sediment, the taro fields were 60 to 90%. And so working with communities there, they were able to get funding to revitalize a number of their taro fields, move them out of the low-lying areas where they were getting flooded by king tides and storms. So for climate change adaptation and sea level rise, move them up into the watershed, and they're even more effective. So now there are 100 taro fields that are in production now that didn't used to be there, and they've been able to use these to cut down on the sediment load tremendously. So there is some interest now. There's a little bit of work going on Maui using the same technique. These could be established elsewhere, but again, this is a no-brainer, culturally acceptable, economically feasible, and it's a really good solution. And then finally, Pohnpei, the upper picture, you can't see it too well, but there's a couple of red zones there, which were mudslides. This community in Pohnpei did a very smart thing. They set up a marine protected area because they knew their fishing was going down. But they have so much trouble. Their cash crop there is called sakau. You may know it as yangona or kava, but that's their big cash crop, and they just love sakau. That's what they do. And so if you haven't had it, it's kind of a muscle reluctant. It kind of knocks you out. I've heard stories. And so they were cutting down the rainforest to grow sakau, and the problem was sedimentation and slumping coming down. And it was the marine protected area they set up wasn't working because of the sediment. Herbivorous fish hate sediment because they can't clear their gills, and they also can't see predators coming because sharks and barracuda use this trick called lateral lines. It's underwater sonar. And so they don't have to see their prey to hit it and eat it. But it's like being in murky water. Nobody really likes it. You know, there are things, you hear that cello music in the background. Dun, 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 dun. And so herbivores weren't there, even though they were protecting the area. So as they began to do the Grow Low program, stop cutting the rainforest and then move the sakau farming low, then when the sediment dropped down, the fish started coming back again. So the connection between land and sea is very clear. And then Maunalua Bay, one of our favorites. So a lot of our work is here. You all recognize that just down the road. And here you can see by law, you're required, if you're going to do construction, you put up your sediment screen. Does anybody think in any way, shape, or form that that upper left-hand picture, that screen does anything? Absolutely not. So you're doing what you're required to do by law. The law doesn't say you can't kill coral reefs or you can't exceed water quality issues. It says you put up a sediment screen. It didn't work and it never will. And so we can see the water coming down the concrete runways. We have one of the most efficient systems in the world for killing coastal reefs of any place I've ever been, with these concrete runways from the mountain to the sea. And you can see the side channel here that fills up every time it rains, and there's a variety of chemicals in there, but it doesn't go all the way to the ocean. It goes through about 7 to 10 evaporation cycles until it's really, really, really toxic. And then a big storm comes and runs everything into the ocean. And the best way of explaining it is that it's not mean values. They say, well, the mean concentration of this chemical or the mean amount of rain is blank. And it's like saying, well, I had 365 glasses of wine this year. If you divide it up into 1 glass of wine a night for a year, if it's red wine, it's supposed to be okay for you. But if you had 365 glasses of wine in a night, you'd be dead. And so when we say the mean value, I don't care about means, I need to know extremes. Every time it rains, it's like 150 glasses of wine in a night, and the reefs die. And during one storm, 2008, I was supposed to go with Jerry Davis up to Hawai'i School. We had a project with the kids up there. The road got washed out because of the rain. So what do? Go up to check out your watershed. That's exactly what I did. And so this is what it looked like. Category 4 rapids at the top of Kuli'o'o. That's what it looked like. And then this incredible rush of water coming down the concrete runway. And during this one storm event, we had 20 tons of sediment deposited by the second mooring buoy in the channel in Maunalua Bay within 8 hours of that rainstorm. No coral reef can withstand 20 tons of sediment in 8 hours. And so you didn't need to have a PhD to figure that one out. And that's why what we're doing on land is really, really, really a problem. The final things I want to put together is that coral reefs the health of the coral reef actually affects a lot of things as well. These are 2 panels showing northern Palau. There's a little cut there called Ebel Channel, which is a grouper spawning aggregation. I was just up there and it's just so cool. And I'm swimming around and there's grouper this big just hanging out. They spawned like a week ago, so they're just hanging out for another 3 weeks until they spawn again. But, just hanging out with big grouper. It's just really cool. They don't care about you. They just kind of come up, check you out. They're like, hey, I'm a grouper. This is a channel. I'm good. And so we did some work with the community there. They were going to turn this one area bay into a large fisheries port, and it would have killed the coral reef there. And so working with a long-term colleague of mine who does modeling, turns out that for the first 2 weeks after a fish egg is fertilized, the fish larvae are more or less at the mercy of currents. They just drift. And at about 2 weeks, these grouper, their spines harden, and they can begin to orient and do swimming in a direction. So how do they know where to go? They do 2 things. They can smell a reef. So there's a chemical signal that comes off of a healthy reef or a dead reef. And they also can react to sounds and vibrations in the water. So things like waves hitting a shore will cause them to orient. The clicking sound of shrimp will also bring them back. And these sounds travel much further underwater than they do on land. So what he showed was that if the reef is dead, you'll get pattern number 1, which is the larvae going out to sea and never coming back again. If the reef smells good, if it's a healthy reef and has the right chemistry, and the reef is noisy, as it should be, then the lower panel shows 30% of those fish larvae coming back in through the channel and refeeding that area again. And so when we met with the community, again, our job is not telling them what they should or shouldn't do, but providing them with good science. And when the chiefs saw those 2 things, I didn't do it because I have a Mac and this is a PC, but it's a really cool animation. You can watch the larvae go in real time. And the chiefs looked and said, no fisheries port. We need the fish. We don't need the port. And so that's why I love working in Palau. You give them data on a Monday. By Wednesday, they have the policy. And by Friday, they implement it. No place in the world I've ever been like that. And the way they do it, this was the meeting we had with the community, long-term friend and colleague of mine on the left, Noah Idion, who was the speaker of the Palau National Congress. The 2 to the right, Yemen Ngolbu and Stephen Victor, were both my graduate students. One is now the director of the Palau International Coral Reef Center. The other one is the director of the Nature Conservancy for Micronesia. And this is exactly as it should be. Palauans speaking to Palauans. These are the chiefs sitting there. And, my job, Holly, scientists, shut up and sit in the back and just nod your head. So it's not only knowing what you can do, but it's also knowing what you can't do. And in partnerships like that, my colleagues from Palau can do what I could never do as an outsider. And it's totally inappropriate for me to do because I shouldn't be telling people what they should or shouldn't be doing either. But by working in partnership, helping them, mostly writing grants and taking care of the accountability. They were able to do things that I'd love to see happen, but I could never do. And so they were able to speak Palau into the community, meet with the chiefs, do it in a culturally appropriate manner. And this was the community that after one presentation, we actually did the one with IRI when we met with the community there and showed them that the cutting of the mangroves was the problem. When we met with them within 2 weeks, they had a full moratorium on clearing and grading of mangroves. And within 2 years, they had national legislation that no mangroves could be cut without a proper response or restoration thing going on. And so where else in the world can you take advantage of cultural knowledge with modern science and get these kinds of outcomes? And that's why I go there a lot. I go to Washington 4 times a year. I go there much more if I can, because you like to see your work put to use and people that are putting their work to use. We're now living in a world of climate change where we're getting these massive regional bleaching events. We've been hit in Hawaii, Maui in particular, 2014, 15, 16, 17, looks like it's going to hit again. When the corals die, they get taken over by algae. We're getting ocean acidification, so it's harder for corals to grow. And then I talked about this Irish potato famine. So we have local stressors that we can control. It's going to be a harder lift for global climate change. But still, even if we do everything right at the local level, we're still going to lose if we don't care about what goes on at the climate level. So one of my last few slides here, this is my entire life on one slide. So I don't know what it says about me, but I was once told that if you can't tell people what you did on one slide, you haven't figured it out yet. On the lower axis, we have the year 1900 all the way to the left, and then everything to 2009 is real data. On the y-axis on the left is percent coral cover. And then from 2009 to the year 2100, we're just modeling predictions of what we think might happen in the world of climate change. And what it shows us, if you look at the line starting in the year 1900 to the year 2009, it went from 80% coral cover to 38% coral cover. This is the mean for 263 reefs in the Pacific, mostly from the Great Barrier Reef, based on real data. So these were real numbers. And it showed over half the coral was lost during this period of time. So starting in 2009, if nothing changed, that's the red line. And again, models, you can look at, well, 2060, 2070, 2080, the decade doesn't matter. What really matters is the slope of the line. And so what we see is the red line is business as usual. But if we start to put in global warming, then we get the yellow line, meaning that we actually get to that death mark much sooner than we would otherwise. If we do the kinds of rehabilitation we talked about, watershed corrections, get our herbivorous fish back again, begin to cut down on pollutants, we have really good data from reefs in Palau and throughout the Pacific, including Hawaii, that when you stop the insult, you start to get coral larvae, you get reproduction is successful, the larvae begin to settle, they begin to grow, you begin to recruit. And so we have this period of time, and these are actually based on real data, where the curve, instead of going down, is now going up again. But the predictions are by the year, we thought around 2050, now it looks like it may be 2030. But if we don't get climate change under control, then we're going to be back to the green and the blue line. Green is just climate change with warming, blue is when we add ocean acidification. And so when I was working with Eric, he's typical physical oceanographer, it's all about the numbers. And so he had yellow, red, blue, and green. And he said, all right, we're ready to publish. And I said, no, I refuse to publish that graph, because what you said is you have 4 roads to death and destruction. And I'm not going to publish that. I said, we need to have something that shows what happens if we get our act together. He rolled his eyes, you biologist. So we did the model with the purple line. And sure enough, if we get pollution under control, sedimentation under control, fisheries under control, and climate change under control, what we leave for our children and grandchildren is 70% coral cover or less than 10%. And as a parent, not just a scientist, there's only one acceptable outcome up there. And I know what we need to get there. We need to get there by doing the things that we're doing for controlling stressors and then getting the recovery. The way that we do that is building local and regional capacity. And so one of the things we do very well at the Pacific Biosciences Research Center is a lot of training for Native Hawaiians and Pacific Islanders, which are the most appropriate people to be working across cultures. And so my buddies from Palau and have 2 Native Hawaiian PhDs graduating this year, hopefully. And then learning how to be better communicators, and part of the blame is with scientists. We haven't done a good enough job of communicating clearly. So there's a bunch of great resources, and I use this when I'm teaching my graduate students, learning how to be a better communicator. There's great books on how to be able to be a better communicator to the general public, how to work with decision makers, how to be able to separate politics. The number one question I teach a graduate course in communicating science, right before Thanksgiving vacation every year, I have a group of students come up to me and says, how do I go home for Thanksgiving, talk to my relatives about climate change? I've got weird Uncle Ned, and I know he's going to go crazy on me. And it's like, okay. So we talk about ways in which you can communicate across these barriers. A really good one on climate change. You can get this downloaded from the internet. It's called the Center for Research on Environmental Decisions. Psychologists wrote this. It has nothing to do with the science. It has the psychology of how do you talk to people about climate change. And it's a brilliant book. You can just go on the internet, look up CRED, Center for Research on Environmental Decisions, Columbia, and you can download it for free. It's brilliant. We just got finished. I spent the last 18 months on the National Academies of Science Committee. We're so far down from where we should be that now we really have to look at interventions. Things maybe 10 years ago we never would have considered as options, we have to consider today. Because when you're down, I was just in Florida a couple times last year. There are good reefs off of Biscayne National Park at 3% coral cover. Their bad reefs are 0.05. And when I said, no, you mean 30 and 5, right? They said, no. 3% is good, 0.05 is stressed. And I'm like, wow. I'm so glad I don't live in Florida. And so we spent 18 months working on what are 23 interventions across 4 categories we could do, and then a decision-making tool to say, just because we can do something doesn't mean we should, but we can't simply, if we just save what was left today, we're so far down on that curve that it's just not acceptable. And again, that's the parent in me speaking. So I'll finish by saying that, how do we get there? And again, food, most people understand food, that when you're making policy and when you're working with groups like I have a tendency to do, science shouldn't be a side dish, it should be a main course. And when I say science, I mean social sciences, biophysical sciences, economic sciences. You also need policy, you need culture, you need all these pieces together. But if making legislation and policy is like being at a buffet table, you need to make sure, just like in the musubi, there's no musubi without rice. You can't make good policy without science. And that's the part in which we hopefully are making contributions. And I'll end by saying, sometimes you need to make partnerships with the ones that are least likely. I have this history with the Army Corps of Engineers, which has been kind of jaded over the years, but it's gotten a lot better. And it turns out that groups that I never would have imagined being able to work with before are turning out to be some of the best partners we have. So thank you.